Hello everyone. Welcome to the first of our KOL Speak series. The topic of our discussion today will be liquid biopsy at an inflection point, its contribution to precision oncology, and BloodPAC's contribution to it. Today we are fortunate to be joined by two eminent personalities who are both passionate about progressing liquid biopsy and have also contributed to it in their own ways. Dr. Johan, welcome to this discussion.
Thank you.
Dr. Johan is a medical oncologist at UAM's College of Medicine. He has keen interest in the areas of biology and bioinformatics. His focus is on the application of advanced molecular profiling for the characterization of genetic-based alteration in cancer cells. Dr. Johan, it would be great if you can introduce your work as it relates to liquid biopsy and talk a little bit more.
Sure. So I am a medical oncologist. What makes me a little bit unique as a physician is I became a physician as a second career. I initially worked in engineering for 7 years for one of the big engineering companies in the US on the East Coast. And during that time also did a graduate degree in computer science. Before arriving at UAMS about 7 years ago, I was at the NCI, the National Cancer Institute in Bethesda, Maryland, for 10 years. And during that time was also an investigator in the Center for Cancer Research. The area of liquid biopsy is very exciting. It's actually been the holy grail for medical oncology for close to the last 50 years. And it's all because we would like to be able to take care of our patients and be able to monitor their disease and provide very rationally based therapies with in the safest possible manners. So I think it's first important that we kind of define what we'll be talking about today regarding the liquid biopsy, since there's a couple of definitions. It could include circulating tumor cells, exosomes, or cell-free DNA. And for the discussion today, we will limit that to cell-free DNA. That has been really the primary focus of BloodPAC recently. And BloodPAC is an organization composed of members from academia, from industry, and also importantly from government. And Lauren has been the executive director and has created a milieu here, which is quite unique and not easily pulled off, in order to get all the participants to cooperate and work together to, what we wanted to really do was to shrink the time of bringing a successful liquid biopsy assay to the clinic to do 10 years worth of work within 5 years. So I would just like to continue with just a couple of more definitions regarding liquid biopsies and what we'll be speaking about today. Even within the area of cell-free DNA, there's some umbrella terms. So you'll see that in the literature as cfDNA, which means cell-free DNA, or CCF DNA, which means circulating cell-free DNA, or ctDNA, which means circulating tumor DNA. And that is what we're really after here. So I kind of think of the other terms as being umbrella terms for circulating tumor DNA. And what this basically is tiny tumor fragments, which have been released by the solid tumor through the process of apoptosis or necrosis. It's released into the bloodstream. The size is quite small. On the average, they're about 170 base pairs that they're eliminated through the kidneys, and they have a half-life anywhere between 30 minutes to 2 hours. So they can be derived from the tumor. They can also be derived from cell-free DNA can also be derived from normal cell turnover. For instance, our GI tract turns over every month. And also following vigorous exercise, we know that there's release of cfDNA from myocytes that die during exercising. importantly also, cell-free DNA is released during inflammatory processes, like when a cancer patient has an infection. So these are very important to understand the origin of the cell-free DNA. And I would just like to point out also that it was initially speculated on in 1948 by Mendel and Mathis in a publication. But then there was kind of a long silence period. Then in 2001, JAHR and colleagues published a paper speaking about DNA fragments in the blood of cancer patients that was published in Cancer Research. So that kind of had a resurgence. And then in 2008, we had a publication by Deal, specifically circulating mutant DNA to assess tumor dynamics, and that appeared in Nature Medicine. And then, just recently this year, coming up right to the present, we've had 2 FDA assay approvals by Guardant and by Foundation Medicine, both who happen to be BloodPAC members. So it has been very recently, over the last 15 to 20 years, quite an acceleration. But the important thing is the specificity of the findings of these tumor shed products allow us now to use it for assay material for precision oncology. And this is where we want to go. We would like to start. The whole point of precision oncology is really to change medical oncology from an organ-based discipline to one that's molecularly based, where we have a very rational treatment plan for patients based on what we're finding from the tumor from that particular patient. So we can have a very rational drug therapy plan brought up. And this is what we've been trying to do. And we now have much better molecular diagnostics due to next generation sequencing. And really, the liquid biopsy is an application of deep sequencing using NGS techniques. That's kind of my opening.
Thank you very much, Dr. Johan. I mean, with your introduction, it was beautiful to hear from you the liquid biopsy and cfDNA specifically, what we're going to focus on today. I think during the conversation, you mentioned about Lauren. So I think half of my introduction is done. So Lauren joins us here. Lauren Lehman, she's currently the executive director of BloodPAC, a consortium that's focused on accelerating the development, validation, and clinical use of liquid biopsy assays. Prior to running BloodPAC, she was the Senior Director of External Partnerships at the White House Cancer Moonshot Task Force. Welcome, Lauren.
Thank you. And it's hard to follow Don.
I think it would be nice if you can talk a little bit more about yourself and BloodPAC's mission before we actually get into the discussion.
Sure. As Don mentioned, BloodPAC was founded back in 2016 as an initial commitment to the White House Cancer Moonshot Project. The idea was to make a decade of progress in half the time. And we kind of decided to do that through 2 kind of pathways. The first one to help generate and create standards among a consistent group of stakeholders in the field, and the other to build a data commons, which we now call the BloodPAC Data Commons, to aggregate data that would support the development of those standards in the field. In 2017, we actually became an organization, our own nonprofit, in February 2017, initially comprised of about 20 different organizations. Today, we have about 45. And we have added what I like to say is a third branch to our tree of the work that we do, which is really stakeholder engagement, which has, for the time being, focused primarily around policy and reimbursement. And as we begin to also dive into the idea of education and confidence building in this technology. And as you heard Don, who speaks so eloquently and is able to explain this very easily and well to most individuals, it is a very complex technology. There's a lot going on. And I think we need to have this technology be accessible to all patients. It really needs to be well understood and confident. People need to have confidence in it. And so that is kind of the third branch that we're currently focusing on as well in BloodPAC.
Thank you, Lauren. Okay, so to start off today's discussion, I think both of you mentioned that it's a complex field, yet we have seen tremendous progress, acceleration in this field in the last decade. So, Dr. Johan, in your view, what are the top 3 factors that have led to this accelerated progress in the field and which has helped liquid biopsy reach its inflection point?
Well, I think that first, liquid biopsy is really seen now as a new source of cancer biomarkers. It really adds a new dimension to clinical trials. It also can potentially address some of the problems associated with what we call solid tumor heterogeneity, which means when somebody has metastatic disease, you have a primary tumor and metastatic foci, we know that molecularly some of those tumors and foci can be quite different. So the liquid biopsy potentially allows us to get a much better sampling from the concomitant nature of the totality of the patient's cancer burden. I would also like to say that with some of the new therapies we have, especially with immuno-oncology, we're now running into some additional challenges regarding medical imaging, where sometimes the immunotherapy actually cause the tumors to appear bigger on medical imaging, because basically the immune cells are being attracted in there and are eating the tumor. So I would say not to replace medical imaging, but liquid biopsies and more advanced molecular diagnostics adds a new dimension and possibly a synergistic type approach, especially for regimens involving immuno-oncology. The other thing is we now have a way to monitor a patient's disease much better. For the most part, when patients are initially worked up, we do a biopsy on their solid tumor, and we very carefully weigh the risks versus benefits of doing that and deciding exactly what lesion to go after. Safety for the patient is always paramount. Now with a liquid biopsy, we should be able to increase the frequency of that. And for certain cancers where we now are using additional techniques like targeted therapy in lung cancer, I mean, thoracic oncologists have to be very nimble on how they treat their patients, because we can get tremendous responses with some of the targeted therapies, but we know over time that resistance will occur, and so you need to resample that new tumor, the new clonal outgrowth, and then revise your therapy plans. So you have to be quite nimble with that. Now, to try to do that with solid tissue biopsies in the past has been quite problematic, because what I said, for lung cancer patients, the average age is 70. They usually have other comorbidities. They may have COPD, they may have heart disease. So we have to think very carefully before we stick a needle into the chest and maybe drop a lung. The liquid biopsy is just a routine blood draw. I mean, the safety profile is just terrific. And so the liquid biopsy really lends itself to more frequent monitoring, and may I think really change the way we think about adjuvant therapy. So adjuvant therapy means, the simplest example would be somebody has a solid tumor, you operate on them, the surgeon comes out and says, I got it all, And we know through some of the big clinical trials we've done that for many patients still systemic therapy will have a benefit. But we also know that we really overtreat people, and sometimes we cause additional problems. A good example of that is Robin Roberts, who's a very famous newscaster who had breast cancer. She was cured of her breast cancer, but then she developed a leukemia. Because of leukemia, she had to have additional therapy and a bone marrow transplant. So we have therapy-related cancers that get caused specifically because of overtreatment. If we had better assays where we could monitor a patient and on an individual basis make a decision whether adjuvant therapy is really warranted or not, that is something that we really need to explore. And now we're developing the tools and methods to do that. The final thing that I would say in this area is just regarding drug development. ctDNA is really a terrific feedback. You can start answering the questions, am I hitting this target? If you have a solid tumor with a certain mutation and you're using a targeted therapy, the assay should reflect whether you're hitting the target or not. If not, because of drug expenses and really financial toxicity, some of these drugs just cost a tremendous amount of money. Like a month of lenalidomide costs $27,000. A month of some of the targeted therapies for lung cancer cost between $10,000 to $20,000. So even if a patient has good insurance, a 20% copay, I mean, how long can you do that? I mean, it's really rough. So I think that when you give physicians tools and allow them to assess the feedback of their therapy, then just great things happen. And a great example of that is just what happened with in HIV and a lot of the HIV medicines, and how the ID docs were looking at viral load and CD4 counts and really customizing therapy based on that. Now we have a way to customize therapy for patients with certain types of cancers with a good feedback and be able to modulate and revise that therapy on a very rational basis. And when you have things like that, when you give physicians those tools, really tremendous progress has a terrific potential to happen and really help people throughout the world.
Very true. Yeah, I mean, liquid biopsy has made longitudinal tumor profiling, which was what's probably a dream true.
Correct.
So Lauren, would you like to add any further points in terms of pivotal factors that have brought liquid biopsy to the place where it is today?
I mean, I think Don named all of them, most of them, if not all. I think I always like to say, Don did mention a lot of the patient factors, but I do think the reality that this technology is, helps patients and their families actually proceed through the testing process in a much easier manner, as Don really described, is incredibly important and shouldn't be forgotten when discussing the actual technology itself. The impact, the ease of this has on patients and their treatment process is incredibly important and needs to be considered throughout, the entire discussion. I just want to always try to make that, kind of bring that to the forefront of the discussion, because it is what we do, and at BloodPAC, I think Don said and I've said, our mission is solely to accelerate the development, approval, accessibility of this technology for patient benefit. And it's a huge benefit to be able to use this.
Thanks, Lauren. I mean, while we are all discussing that it has accelerated, there has been tremendous development, but I think somewhere our point that we are working towards making it come faster to the market, validating it better, all of these say that we still have quite a bit of ground to cover. So Dr. Johan, what do you think are the key challenges faced by clinicians in being able to adopt liquid biopsy as routine cancer care resource? Is it like you were mentioning that it's deemed suitable for only certain types of tumors? Or is there any concern about comparability with tumor biopsy results, or anything else?
Yes, so those are really great points that you bring up. One thing that we've been very cognizant about at BloodPAC is, kind of avoiding overhyping this. Sometimes, you have a tremendous technological advance, and then you start thinking of some great applications of that, and you start to see the semblance of tremendous movements. However, you still have to be quite careful about this. You don't want the technology or the methodology to get a black eye. So because it's being pushed out too fast, and you have this irrational exuberance about it. So I think that education is a very big component to this. So what happens at academic medical centers is quite different. What happens in the community, and on top of that, there's also geographic disparities. What happens in Boston or New York City or Bethesda, Maryland, is quite different. That may happen in different parts of the world, or even different parts in rural Georgia, or a place like that. It's quite different. So it's important to kind of bring this out to everybody, and that's just been a big problem as well with precision medicine in general. The other thing is, as you brought up, is comparing it to the gold standard, and the gold standard is still tissue biopsy. And because of that, you have to look at, I would say some of the things just regarding accessibility. So I've spoken to that a little bit more. When a person is diagnosed with cancer, there's sometimes accessibility problems with either getting some of the solid tissue from an outside facility, or repeating that biopsy, and that may require a big medical team, surgeons or interventional radiologists, anesthesiologists, pathologists, versus the liquid biopsy, where it's just a routine blood draw. And then on top of that, then would be the turnaround time of comparing solid tissue analysis versus liquid biopsy. To schedule all of that, different medical specialties, and to schedule a small operation or an invasive procedure is quite different from just doing a routine blood draw, maybe using a special tube like a Streck tube, and either sending it out or doing it within a CLIA lab. The sensitivity of these assays, solid tumor versus the liquid biopsy, is quite different. The sensitivity in the solid tumor assay is really excellent because all of the biopsy samples undergo a review and assessment for tumor cell count and purity. And genotyping would then only be performed on specimens deemed adequate for analysis, where with the liquid biopsy, it's going to be a bit lower, since there is no real suitable review. For instance, we can assess tumor cell count or purity, and then depending on the tumor type and the burden of disease, the ctDNA may not be detectable, particularly with tumors that are known to have minimal shed. The specificity comparing solid tumor analysis versus liquid biopsy is also quite different. Usually the specificity for a solid tumor is excellent, except sometimes you may have germline variants reported as somatic. This can be resolved if the germline sample is included in the analysis, and you can subtract that out bioinformatically. The specificity for liquid biopsies is really excellent for targeted driver mutations. We do get false positives again for certain genes, especially at very low allelic fractions. And this can again be due to germline leakage in, specifically a phenomenon that we're now understanding much better, called clonal hematopoiesis of an indeterminate potential, or CHIP. But again, we can address that by including germline, from the buffy coat into the assay, and then bioinformatically subtracting it out. The expense we've already spoken about. Certainly to schedule a small operation or a procedure with multiple medical specialties can be quite expensive versus a routine blood draw and just sending it out. So that's quite different. Something that you brought up regarding longitudinal or the repeatability of these assays. For solid tumors, we normally don't repeat. We'll work up a patient, we'll start them on a therapy, and after 8 to 12 weeks, we'll bring them back, we'll repeat the CT scan, see how they're doing. For the liquid biopsy, we should be able to repeat this. As the price of next generation sequences continue to fall, and it's been quite dramatic, it's been outpacing Moore's law now for years. The hope is really that the price of these assays will come down so that we can repeat them more frequently and really help people a lot better. And then, as I just mentioned, initially has to do with the scope of the analysis that the assay can perform. With a solid tumor, you're always worried a little bit about spatial bias. Where exactly did that needle go in and what am I seeing? And when you look at a solid tumor, you can see, all sorts of things in the tumor, meaning low-grade disease versus very aggressive disease. And again, it depends on where the needle went in, how many core biopsies you took, and the potential for liquid biopsies is it should be able to potentially to sample from multiple foci, so you would get a much better idea of the scope of disease and be able to design a much more thorough and rational therapy plan for a patient.
So, sounds like a combination of both clinical and non-clinical factors that are affecting a clinician's choice.
Yes.
Yeah. So, Lauren, while there are several challenges that Dr. Johan just mentioned, BloodPAC is working towards alleviating some of these concerns. So could you talk to us about a few projects that have helped clinicians gain clarity when it comes to some of the challenges? Dr.
Sure.
Johan.
Absolutely. And I'm also happy to talk about, I think what's interesting about BloodPAC is not that we're only helping clinicians with making the best decisions for patients, but I think what our aim is to really help the development of these diagnostics, starting from the very beginning, so the developers to kind of accelerate their time frame and their development of these, helping then obviously clinicians have access, understand what it is, researchers understand, have regulatory agencies like the FDA and hopefully soon to be other external global regulatory bodies, help them understand and create some standards for them to be able to review these tools consistently, and in a way that we've worked together to kind of bring those that understanding to them, so the review process can take place faster. To then actually being in the clinic. And, one of the things that Don just mentioned, the price is coming down as we, get farther and farther along in this process to run this technology. However, we're aiming to have this access for all patients. And that really requires reimbursement and payers to kind of understand the benefits of this technology, and we are working in that area now as well. So specific projects that BloodPAC is working on. Don is actually a leader of multiple of our working groups, We started off very early on, even back during our time as a White House Cancer Moonshot Project, reviewing and understanding pre-analytical variables that will impact the outcome of the assay. Very simple things like, what tube type are you using? And we go through a very simple process, I think, but it's kind of very regimented. It's iterative. We work together among all of our consortium members to come together and say, here's our practice, here's what we do, here's our protocols. We aggregated in that specific project a group of about roughly 50 different pre-analytical variables that different companies and organizations felt were really important. We whittled them down to 11 core variables that we thought were essential. We presented them to the FDA, who then said, well, I'm pretty sure there's a lot more of these. Why don't you go back and take a look? Which we did. We then went back with the rest of the group, reorganized, said, hey, let's look through all of these. What do we think is really critical? And when I say critical, it's important to understand that I'm saying, what are these minimal technical data elements that we should be requiring for submission of data to the BloodPAC data commons? It should be acknowledged that we are slightly different consortium because we are made up primarily of corporate, businesses and not just academics. We have a few wonderful academics who partner with us. We have a few fantastic foundations that partner with us, but the majority of our membership are companies. So we need to be clear about what data they have access to, or we shouldn't be asking for it if they can't submit it. And that one project, ultimately, we were able to speak with the FDA multiple times and whittle those numbers down to 11 of those core variables that we felt were critical. And we were able to get support from the FDA for those 11 pre-analytical minimal technical data elements. And then we went to CAP, the College of American Pathologists, and they too lent their support for those 11 pre-analytical variables. And we actually have a paper that was published back in April of this year on those 11 minimal technical data elements around pre-analytical variables. We have continued on, and Don is actually the co-chair of our clinical variables working group. We have a clinical variables working group and a patient context variable working group, looking at variables and impacts that revolve around the patient and variables that revolve around that are disease specific. And what are the minimal variables in those two areas that we can, again, standardize and require for collection when you're looking to submit your data to the BloodPAC data commons. And hopefully shortly, and then by the end of the year, we'll be able to provide that list to the FDA and get some feedback and see where we stand on the creation of those. Also in that process, most recently, we published a paper through our Analytical Validation Working Group that is led by Jim Godsey from Illumina, Angela Silvestro from Novartis, and John Baden from BMS, that focuses on the creation of a generic analytical validation protocol. That document is that was published in its entirety in September of this year. And Don is an author on that paper as well. Really outlines all of the protocols, and it's the culmination of work for 2.5 years from a group of about 15 different organizations and individuals who came together, who walk step by step through protocols that should be used for the validation of their assay. And we worked hand in hand with the FDA to create those. We went through 4 official pre-sub processes with them, multiple offline conversations, and I would say roughly 60 to 70% of the language in the actual protocol itself is probably directly from the FDA. I encourage people to go look at it, review it. Our intention is to have as much feedback as possible. It should not be a static document. It should be something that is kind of given out to the larger liquid biopsy community to say, this is what today the consortium of a total of 46 organizations have agreed upon. There are a lot more people out there, and so we hope to elicit their feedback. As a result of that publication, though, the FDA has requested that we pull together a kind of version 2.0 of the generic protocol that will include MRD. There is a working group right now that is working on creating that version 2.0. I anticipate it'll actually be ready for an initial pre-sub probably in early spring of next year. And there is a small splinter group of individuals who care a lot about blood TMB, and they are working also in kind of an appendix to the first generic protocol to include blood TMB. So again, those are creating standards for the community to utilize, standards for the FDA and hopefully other regulatory agencies to say, hey, this is actually a really good guideline, and at least 45, 47 different organizations have agreed upon this. How can we help use this to accelerate the field? In addition, very quickly, we also have kind of in that data realm, we have a working group that's focusing on looking at the question of liquid tissue concordance in lung cancer. And how do we, as a data commons, become a source of valid scientific evidence for the community by using our data commons? We're going to be partnering with the FDA in hopes of becoming kind of supported by them to answer this question. We've collected data from Don and several others in that to kind of focus on that initial question. It's really a demonstration project, and I think it's we've made really big strides in the past few months. I think our initial, I'm looking at Don, I think our initial numbers were going to be 1,000 different patient samples, and I think we're up to closer to 2,000 to 3,000 patient samples now that we've received from multiple different partners. And then in kind of the space of kind of stakeholder engagement, we recently, very early on, I'd say at the beginning of our founding as an independent organization, we brought CMS and a couple payers to come speak at our meetings. And it was very important to hear kind of their thinking on reimbursement. But at the time, there just wasn't a whole lot for us to do. About a year ago, as a community, as an entire consortium, we decided now is probably a really good time to start having a more in-depth conversation about where reimbursement is going, where policy is going, how BloodPAC can be involved. If we're focusing on how to accelerate the development and approval of this technology, we really should add a column of the accessibility of this technology. And that really boils a lot down to reimbursement and education, and again, building confidence in this. And so that working group focuses on addressing those issues. And I would say, I'll stop talking. I would close up by saying, I think that there are two primary reasons why BloodPAC has been so successful. The first really has to do with our structure. We work in these very, intimate working groups. There are about 10 of them that are structured right now that focus on very specific projects. They move those projects forward at a very rapid rate. We're not trying to solve world peace. We are simply trying to address these small bite-sized chunks and issues, and then move on to address the next issue together collectively. And I think that's been a really successful model for us, and I encourage others to use it. And then the second is, you're really lucky. We're all really lucky to have Don participate in BloodPAC, to have Don speaking here. And there are 120 individuals that are partners within BloodPAC that represent the 47 different companies. And they are truly passionate about this technology, but more importantly about giving patients better options and having better outcomes. And that you couldn't ask for more to run a consortium and an organization to have people committed to that. And we're really lucky.
Sounds like you have a great team working with you.
We do.
Yeah. Okay. So I think we've discussed quite a bit of where we are right now and our current projects. But I think part of all conversations is to see what's in store. So looking at the crystal ball, Dr. Johan, what do you think are the key factors that are going to drive the field of liquid biopsy and help it establish itself as an important part of precision oncology?
Sure. So in a nutshell, it's all about clinical validity and clinical utility. Liquid biopsy, for all of its accolades, is not really proven yet. There's still issues with really NGS-based oncology assays in general. Really, do NGS assays and NGS-based assays on blood now, which is even harder than tissue, does it improve patient care? We want to use the liquid biopsy to guide personalized therapy. Now, normally we'll start out and we'll do a retrospective examination, but eventually that has to be moved into prospective evaluations as well. And so, as we mentioned before, another key point of this is longitudinal type testing. Specifically, that means repeat testing. Is the patient responding to the drug? Again, we have currently no clinical validity or clinical utility regarding this. And just to, again, define something, when we're speaking about clinical validity and utility, really it's a clinical validity really is the ability of an assay to divide with statistical significance one population into two or more groups on the basis of outcomes, like for instance, treatment response or presence of cancer. This really hasn't really been done yet for the liquid biopsy. And then clinical utility would be just further along with that, demonstrating again with statistical significance improvement in the diagnosis, treatment, or management or prevention of cancer with the use of the assay versus not using the assay. Again, with liquid biopsies and really with a lot of NGS type testing, this has not been done yet, but we are getting closer and closer to this. You just don't start out trying to do this. Again, the assay gets rolled out, it starts to be used in clinical trials, we start to do retrospective examinations, and then we start tuning things, and then we do a prospective analysis. So this will be, these are the final clinchers that we're all working towards, that we know are the target that we need to hit. And we believe that we have a good projection in order to hit them. But again, it takes a group effort. You need clinicians, you need scientists, you need great administrators. And so we think we have a team composed of that. The other thing is that, along with this technology comes something new to biomedicine, and it's this tremendous amount of data and all of the quantitative techniques. So again, BloodPAC is very fortunate to have somebody like Dr. Bob Grossman from the University of Chicago, who is an expert on constructing data commons. He's probably the best in the world. He's a member of BloodPAC, and we really need this type of infrastructure in order to collect the data, to harmonize the data, and then perform the appropriate analyses to answer these type of questions.
Thank you. So Lauren, to sum it all up, while there are several factors that Dr. Johan mentioned, is there a message that you would like to give assay manufacturers, industry, academics, patients, all different types of stakeholders, payers, everyone including, what is it that would help you catalyze and help liquid biopsy become more a prominent resource in cancer care?
My incredibly non-scientific answer is the one I started with, which is collaboration, right? Obviously, we started BloodPAC almost 4 years ago with the idea, after reviewing numerous documents and reviewing, speaking with so many companies and researchers, I finally turned to my colleagues at the NCI and the FDA and said, this really isn't a science issue. This is really a collaborative issue, that if people were willing to come together to share their standards, to share their pre-competitive knowledge, to share their data, we would be able to accelerate the pace really quickly. And I think that's been done. All the points that Don made that, at the beginning of this talk really validate, I think, that BloodPAC has helped both the two companies, Foundation and Guardant, both are members of BloodPAC. And, where we started 4 years ago, they've made dramatic progress, and they are active participants. And I think, my wish is to see, not just in liquid biopsy, but collaborative efforts and consortiums built around in multiple areas where you're really able to take the broad diversity of stakeholders in the field, bring them together, use their incredible knowledge and experience, and use that as a catalyst to accelerating the pace of discovery and driving these issues forward for patients. I think that's the reason why, it's my personal reason why I think we've made such a tremendous amount of progress so quickly in this field. And again, I hope BloodPAC is used as a demonstration project for the broader, healthcare field and beyond to show that this is possible. But Don, maybe I think it's important to hear also from you, your opinion on that.
No, I think that, again, it takes it's really a multidisciplinary group effort. You need a very good people. Administering a project like this is extremely challenging, especially like Lauren said, we have a tremendous amount of industry partners involved. We have representatives from all of the big pharma, from biotech, Illumina, Thermo Fisher, and to get people working together for the greater good, it's much easier said than pulled off. And it's really, Lauren who's been able to pull this off. I'm basically a lab guy. That's what I love. And, but to be involved in something so practice changing is really where the reason why I went into medicine. And so I just feel really fortunate to be involved in this and to start to see these, the successes through the publications, through the meetings, through the progress that we've made over these last 4 years. It's really been remarkable.
And I will add the plug. We're open to others joining us. We are a extremely inclusive body, and I think we're both well aware that there are huge strides being made globally, both in the area of development, in clinical practice, and regulations. And I think it's really important to bring the entire global community forward, and that's really important. And so I've received over the past month actually 4 requests to participate in BloodPAC from outside the US, which is extraordinary, and I'm thrilled about that. And so I will make the plug here and now that we'd love to have participation from others, and looking for experience and interest in people collaborating together to kind of push this field forward.
Yep, definitely. Looks like the world is coming together.
Yep. I mean, cancer just doesn't affect the United States. We're very well aware of that. And these results, we would like to, everybody to benefit. Yeah. Everyone, I grew up in the computer industry, and I used to see, again, Moore's Law. Why can't we apply that to biomedicine? Why is it that we can't start cutting the prices of molecular diagnostics and drugs and all of that the same way it happened in the computer industry? I mean, it all starts first with having the will to want to do that, and then putting the right people together to make it start happening. And nobody ever thought, like when you look back and you look at what was going on in the 50s and the 60s, that where we would be now with the way, you don't have 5 or 6 computers in the world. I own 5 or 6 myself, and they're high-end machines, and they don't cost that much. And why can't we do that for healthcare and cancer medicine? That is a question that I'm constantly thinking about. And I think it's with these first you have to have a technological breakthrough. And we've had that with next generation sequencing and high throughput technologies. And then you start having some good ideas. And then sometimes wonderful things start happening. And being able to bring this to everybody is what we want to do. And really another thing that makes BloodPAC such a special organization. And it's really because of Lauren and her leadership. I mean, she's the executive in charge.
Yes. Good things happen to good people. So I guess that's why everything is working. Okay, so any other closing thoughts?
Thank you for having us.
Thank you so much for
Thank
agreeing
you.
to participate in this discussion. Thank you for sharing your insights and helping Course5 understand liquid biopsy from your point of view. We wish all the success in helping liquid biopsy find its true place in oncology. I think ultimately patients finding better results. Thank you.
You're welcome.
Thank you.